So we will be doing today's talk on an update on the Sikuliaq research cruise that was occurring in November, if I believe that's correct, right when we were having our previous Arctic survey readout. So we're really excited to have Seth and Jackie here to be able to present on not only that cruise, but also I believe some of the other results from their work this summer. So before we get started, I'm going to suggest we go around the room and do a round of introductions since we're a smaller group today. So if you just state your name, affiliation, and if it's your first meeting with us here at IARPC, we'd love to hear that as well. So once again, I'm Andrew with the IARPC Secretariat and NOAA Fisheries. And I will just kind of go down my list here. Danielle.
Hi, I'm Danielle Dixon with North Pacific Research Board in Anchorage, and I'm a co-lead of the Marine Ecosystems Collaboration Team. Welcome, thanks for joining us.
Thank you, Danielle. Seth Danielson.
Morning, everybody. I'm Seth. I'm a physical oceanographer here at the University of Alaska Fairbanks.
Thank you, Seth. Kathy.
Yeah, good morning, everyone. I'm Kathy Kuhn, Bureau of Ocean Energy Management, and also one of the co-leads of IARPC MECT with Jackie and Danielle.
Great, good to see you, Kathy. Meredith.
Hi, everyone. Meredith LaValle. I'm with the IARPC Secretariat. I help to manage the several of the IARPC collaboration teams, and I'm joining in to listen to what the Marine Ecosystems team is doing.
Great, thank you, Meredith. We have Laura Grebmeier. That's okay. We can move on. Kelly.
Hi, my name. Yeah.
Sorry. Oh, sorry. Go ahead. That's okay.
Sorry, there's a delay here on my end. So my name's Laura Grebmeier. I'm a PhD student with Lee and Jackie at University of Maryland, and I also work at U.S. Geological Survey.
Thank you, Laura. Next, Kelly Monteleone.
Hi, I'm Kelly. I'm an underwater archaeologist at the University of Calgary.
Thank you, Kelly. Christina Bonsall.
Hi, everyone. I'm a marine ecologist with the Bureau of Ocean Energy Management.
Thank you, Christina. Lee Karp, boss.
Hi, I'm Lee from the University of Maine. I'm a biological oceanographer. I'm sorry, my camera is not working. And I've been in one meeting in the past. Unfortunately, I was not able to join the meetings, but I hope to be able to start and join in the coming year. So.
Well, we're glad to have you back. Thanks for joining. One sec, I think I lost my place in Lisa Eisner.
Yeah, hi. I'm excuse me, excuse me, haven't talked much today. A biological oceanographer at the Alaska Fisheries Science Center in Seattle, and I've done a lot of work in Bering Sea and Chukchi.
Thank you, Lisa. Greg Anderson.
Hi, everybody. I'm Greg Anderson. I'm a program officer in the Arctic section at NSF, and mostly just interested in hearing today's discussion. So, thanks.
Great. Love the background as well. Next, I believe, is Yvette.
Yeah, I'm from Oregon State University, College of Earth, Ocean, and Atmospheric Sciences. I'm a physical biological interaction modeler.
Thank you. Gail Ashton.
Hi, I'm Gail, a marine biologist at the Smithsonian Environmental Research Center based down in San Francisco.
Excellent. Jackie Grebmeier.
Hi, Jackie Grebmeier. I'm a biological oceanographer at the Chesapeake Biological Lab at University of Maryland, and I study pelagic benthic coupling.
Thank you, Jackie. Hazel.
Hi, everyone. I'm Hazel Shapiro. I'm also a part of the IARPC Secretariat, and I work with the observing data and modeling teams. And it's nice to have a little bit of a quieter week and be able to drop in and listen to this meeting. So I'm looking forward to it.
Great. Thanks for coming, Hazel. Christina.
Hi, I'm Christina. I'm actually just finished up my PhD with Jackie and am getting ready to head off to Iceland to teach for a semester. So I do benthic ecology and pelagic benthic coupling as well.
Very cool. That's great. Candace Nachman.
Hi, everybody. Candace Nachman with NOAA Fisheries Office of Policy, Senior Policy Advisor and the NOAA Fisheries Headquarters Arctic Liaison, and also the co-lead of the soon-to-be disbanded Environmental Intelligence Collaboration Team at IARPC.
Yes, thank you, Candace. And Kathleen Vestfalls.
Hi, I'm Kathleen Vestfalls, and I'm a research associate at Simmers at OSU and the Alaska Fisheries Science Center.
Great, thank you. Is there anyone else that I missed? Feel free to introduce yourselves. If not, I think we can hand this over to Jackie Grebmeier, who is going to introduce today's topic and introduce the presentation that she'll be co-giving with Seth.
Sure. Thanks, Drew.
All right.
Yes, great. It's great. I just got back from Texas, so that was a nice offline trip, but it's back in the real world again. We're going to talk about the Sikuliaq cruise as part of the Distributed Biological Observatory, the Chukchi Ecosystem Observatory, and the Arctic Marine Biodiversity Observing Network. All of them are embedded into some of the performance elements on the Marine Ecosystem Collaborative Team. And I would just point out, if you all don't know, we're in a new 5-year program within the Interagency Arctic Research Policy Committee, and there you can read all that report on the websites. And I think there's another debrief that's going to happen pretty soon about the 5-year plan. But anyway, we cross over in multiple disciplines in the marine ecosystem team, including human co-production of knowledge issues related to food security and activities in the Arctic. So today's discussion, Seth Danielson, he's a professor there at the University of Alaska in Fairbanks, and he was the chief scientist on the Sikuliaq cruise that we had in November of this year. So a late season cruise, quite interesting, and I think that's what we're going to present on. And I'll also be doing he'll pass the baton over to me saying during the talk. But at that point, Seth, I think physical oceanographer, I'm a biological oceanographer, and I think we're ready to go. So Seth, do you want to share your screen?
Sounds good. I will do that right now.
Is that real-time snow?
Not quite. Sorry. It's a whole lot darker than that outside right now.
Yeah.
It does make me think that I'm going to have to be snow blowing the driveway. I've snow blowed the driveway twice in the last 3 days, and I think I'm going to have to do that again by the weekend given the forecast. So let's see. At this point, can you see the slideshow?
Right, and you just moved it into presentation, so that's great.
Okay. Perfect. Well, thanks everybody for taking some time out of your week to join us for this talk. It's really fun to be able to share with you what we saw and were able to accomplish. As you may or may not know, this cruise came together in a fairly rapid fashion after another cruise was sort of COVID canceled earlier in the year. And so we were able to reconfigure. We realized that Sikuliaq had a little bit of time in her schedule, and we're able to pull this off in really a fairly short amount of time. The cruise as a whole had participation from 4 different groups, and these groups included primarily the Distributed Biological Observatory. This is where the vast majority of the ship time came from, was through NSF. But we also had support from the funders of the Chukchi Ecosystem Observatory that we call CEO, funded primarily by AUS and NPRB. The Arctic Marine Biodiversity Observation Network, or AMBON, funded through a NOP program that comes through the NOAA IUS office. And then finally, NOAA's Ecosystems and Fisheries Oceanography, the EcoFoci group, and the NOAA Marine Mammal Lab. So we all had participation on board, and in the the culture of doing research in the Arctic, it's a highly collaborative type of effort. And it was a special time to be able to get out there. It turns out that there's not a whole lot of cruises that have focused on the Chukchi Shelf over the last, well, pick your time, 70 years. While we were out there, I dug into the archives and found that there was a cruise in October and November on a NOAA vessel in 1960, and turned out that Knut Agard wrote that results of that up for his master's thesis at the University of Washington. And after our cruise, I wrote to Knut, and he emailed me a copy of that, or actually stuck it into the mail. I got a physical copy. So we can compare some of the measurements that we're making to observations that were made in 1960. But after 1960, there was not another cruise in the Chukchi until 2011, when Karen Astian had the Healy up there. And then there has been a couple of cruises since then. Bob Pickard was on the Chukchi Shelf in, I think, 2018 and 2019, two very different years than we had here. Whereas this year the ice was coming in fairly quickly. He was up there right at the peak of these very warm years of the recent half decade. So this is what we were planning on doing as we were heading out. And maybe Jackie, I'll turn the baton over to you here for a moment.
All right, thanks, Seth. Yeah, so this is the cruise you can see on the right there. Those are the DBO regions for DBO. I know he's using my pointer, but the DBO 1, 2, 3, and 4 are with the dotted boxes around them. And you might wonder why the DBO 2, 3, and 4 are yellow and the DBO 1 is red, because we didn't think we'd make it down to DBO, but the fair weather and efficiency of the ship and the hard work of the team got us down there to do that also. So it was great. So the role of the DBO is to look at ecosystem status and change, and it's actually common through the CEO as well as the Arctic Marine Biodiversity Program, all which were components as well as ecofoci. So as far as the standard measurements, we look at the conductivity, temperature, depth, using a rosette to collect water samples. Another core part of this was to turn around some of the moorings, the CEO mooring up by DBO4, the M8 mooring at DBO1, and also some up by C12. You can see the names on there. And some of these were also marine mammal moorings. Water column collections were the core ones, and some additional ones from the DBO. We had chemistry, nutrients, oxygen, chlorophyll. The eDNA and the harmful algal blooms were one additional measurements that were supported by both NOAA and the Ambon program, which was great. We did that both in the water column and sediments. We looked at zooplankton abundance and biomass. We looked at macrofauna population structure. We're also looking at carbon, nitrogen, chlorophyll, grain size. And we're able to also do some of the HAB, because the harmful algal blooms, some of the Alexandrium form cysts, and they are in the surface sediments. And so we were able to do those collections. And then we had a limited marine mammal survey on the from the bridge. So you can see where we were able to work in that period of time. We started north because the ice was coming back in, and we did work in the offshore of the DBO4 and ice, and Seth will talk about that, as well as in DBO3, managed to get south actually about 4 days before it really iced up over. So with that, Seth, I'll pass it back.
All right. And I should mention that we were not staffed for 24-hour operations in terms of having multiple teams out there to be able to work in both day and night. And so everybody knew going into this, when we got to a line for hydrographic stations or for mooring work, it was basically now or never to do it. We didn't have the ability to wait for daylight for moorings, or to sort of build in the rest time that you often typically would try to do. And this was just purely it was a short cruise, right? Only 10 days. And so we did whatever we could to pack results into those 10 days. And at the end of it, we wound up with one incredible 60-hour push with a little break of about 9 hours as we transited between the DBO1 and the DBO2 regions. But because of that, and we were fighting the weather, at that time as well, with a huge storm coming in from the southwest. But the science team really did a phenomenal job in pulling together, helping each other out with sampling as needed, and taking a lot of half-hour cat naps between stations when the opportunity presented itself. So here's what the ice was like when we just started out on the 7th on the left-hand side, and then as we were getting back into Nome on the 16th. So the Chukchi Sea really went from about, I don't know, you can sort of average by eye here, maybe 40% ice cover to 70% ice cover, plus or minus. So it was a pretty special time of year to be out there, just as this transition was happening from open water into ice cover. As we got to the northern portion of our sampling region, this is the TSAR image that we were working with in terms of navigating, and it was a pretty good representation of what we found while we were out there. Our DBO 4.1 site was in open water, but we were in ice by the time we got to about DBO 4.2 or 4.3. Much of the ice that we were in was relatively light, and that was fantastic because our transit speeds were able to maintain fairly high rate of transit. But by the time we got up to DBO 4.6, we were down to about 5 knots or so, which was one of the big question marks as we were planning for the cruise, is how fast would we be able to work through the ice. And of course, the slower we go, the less of a chance there was for us to be able to get down to that DBO1 section at the end. After the DBO4 line, we went down to the DBO3 region, southwest of Point Hope. We were in sea ice the entire time. This image was already a day old or so by the time we got down there. And by the time we were halfway through the line, again, this ice was fairly thick. This was a big tongue of ice that had advected east into the U.S. waters from the Russian coast, both south and north of Bering Strait. As we were heading north from Nome, we made just fantastic time. Very thin, young ice. We were able to stay west of that more consolidated, blocked up ice that was in outer Kotzebue Sound that we were a little bit worried about as we were leaving Nome. And we drove through a lot of different flavors of ice, lots of little pancakes just forming, frozen over pancakes. Again, this is ice that Sikuliaq moves through very nicely. Here's what the ice was like in the region of the CEO mooring up in the DBO4 region. So anywhere the near surface temperature was shown as dark blue here during the cruise, we were pretty much in sea ice during that time. So you can get an idea that we were in sea ice maybe around 30 or 40% of the time through the course of the cruise. But still a fair amount of heat also is notable here in the northeast Chukchi Sea, as we were leaving Nome, south of Bering Strait, and of course southwest of St. Lawrence Island. The surface salinities, so these are all from the thermosalinograph on Sikuliaq, showing relatively high salinities in Chirikov Basin and along the DBO1 region, and actually fairly fresh salinities averaging 30-31 north of Bering Strait. And then here's what the nitrate sensor was showing us. Not unexpectedly, some fairly high levels of nitrate in Chirikov Basin, southwest of St. Lawrence Island. But even through much of the Chukchi Sea, we're not sitting down there on 0 micromolar. A lot of the places we were had non-limiting levels of nitrate right up near the surface. This water is drawn in from about 6 meters depth. Catherine Burchak was conducting marine mammal observations from the bridge she remarked on really how many marine mammals she was able to see. Of course, much of the time it was too dark to be able to conduct a marine mammal observation. So the little black dots indicate where she was able to have survey effort, and then the red circles give a little relative proportion of the number of animals seen along those track lines. So both Arctic whales, subarctic types of whales like the humpbacks, many pinnipeds. I think the final count was around 2,000 walrus that Catherine noted. So the walruses were heading south as they do as the ice is coming in. And then there were a few other notable observations. One fairly large jumping fish in a little pool of water in what was otherwise pretty dense and fully iced over water. There was one young dead humpback whale, floating bird that unidentified, and a carcass that was lying on some sea ice of a seal that had most likely been eaten up by a polar bear. Okay, so then we're going to walk through some of the hydrography here. And Jackie, do you want to start with this?
Sure. So like I mentioned, we were up on the DBO4 area. So this is in the northeast Chukchi Sea. And what you're seeing are the ODV, the hydrographics of the upper left is the temperature, but you're seeing vertical profiles now. And on the right is the salinity. And you can see that their salinity is higher on the bottom, as Seth was saying, and then the fresher. This is the ice was on the surface. A lot of what was on the offshore areas. In the middle graphic above the map, which you can see, each one of these maps will have a red box around them, and you can see which one of the lines we're on. And if so, then you're looking at the density. So that's the temperature, salinity, offshore being that deeper water, the Anadyr or the Bering Sea water coming that you can see on the lower part of that graph there. Beam attenuation, that just gives you how much light gets through the system. And then you can see the oxygen levels. So on the surface, they're quite high, up around 350, 380 micromoles per kilogram. And then if you look at the lower right one, you're seeing fluorescence. And although it's low pretty well all over this, you only have about 5 hours of sunlight there, but you do see variability from the near shore to area offshore regions. You can see from that. Next. As you go down to the DBO3, now you're in the southeast Chukchi Sea, and you can see the modifications. Again, temperature on the left, but still warm. It's above freezing on the bottom, so you haven't had that full turnover production and brine formation that occurs when the sea ice comes. So it's pretty, like Seth had mentioned earlier, there was still heat in that water. Salinity, you can see that offshore saline water, the Anadyr water coming on the lower left of that graph influences that density you're seeing. And as you head to the, what you're looking at to the right of that graph is getting into more of a coastal water. I'll just jump down the oxygen. Again, quite a bit of oxygen in the upper water column, but a lot lower on that offshore waters to the left. I would say that there's influence both by the water mass, but also a lot of consumption going on by this really high levels of clams that are taking up a lot of that oxygen on the left-hand side of that system. If you look at the fluorescence on the lower right there, you can see there's a little bump up right in that at about a 25, 30 meter depth. So there was enough nitrate there to have some production going on, enough light, or it's settling organic matter at that point. Next, it's your turn.
Yeah, so as we were getting down to the DBO3 region, this is some of the ice that we were driving through. You see lots of small pancakes and just starting to consolidate together, but there's not so much ice that the swell isn't still propagating through it. We drove through this region of ice that was forming, and I wanted to show you a couple pictures of Frazzle Ice. So this is the Chukchi Sea turning into a slushy near the surface, and to me these pictures are really fun to look at because, for one, you're missing all the capillary waves. All that we're seeing here are longer gravity waves, but the Frazzle Ice crystals, you can see the sun glinting off of the crystals. It's not that the photograph is such a poor quality, it's that really the water is becoming granular as the ocean is freezing up. Another view of this looking into the sun looks like this. And again, you get a sense for that really granular character of the ocean and the complete lack of the small capillary 1 to 2, 3 centimeter waves that the wind typically steers up. So this was all kind of neat because we also had an EK80 fisheries transducer system on board with 5 different frequencies. And we think that we were able to also map out the presence of Frazzle Ice. So one of the things that we do is try to understand the habitat and the partitioning of the habitats. And
Seth, I'm seeing a white screen. Is there
yeah, that's on purpose. I was just going to talk.
Sorry.
Yeah, thanks. So we've got a set of pictures here. Here's our slide of one shot of the EK80 data. So low frequencies here on the left-hand screen. There's 5 panels. Low frequency, 18 kilohertz, 38 kilohertz, 125. Oh no, maybe there's a 70, 125, and a 200 kilohertz over on the right-hand side. So in the past, we know that signatures that look like this in the acoustics, you can see the seafloor at the bottom, that's shown in red. And so the seafloor repeats itself across each one of these little time series, which might be, I'm not sure, half an hour or an hour in length. So some sort of zooplankton is creating the backscatter on this right-hand side. These are elevated backscatter returns in the higher frequencies. This could be euphausids, they could be copepods. In some of the stations that we were at, we saw the signature of what looked to be fairly large fish. I'm not an acoustics expert, but I have passed these slides by some people who are, and their interpretation was the same as mine. It looked like there's some fairly large fish occurring in the water column. Interestingly, the fish all seem to be hanging out below about what is, I think, about 20 meters depth. And those fish are sitting in what appears to be, I'm going to back up a couple slides, this relatively warm layer that's sitting below the cold layer at the surface. So it was a little bit unusual. I hadn't really expected to see warm water sitting below near freezing water, but the salinity stratification was strong enough to keep the water column stratified. Sea ice was present across the entire DBO3 line here, but there was temperatures that were 2 to 3 degrees above the freezing point in the lower half of the water column, and that occurred both on DBO3 and a portion of DBO4 had warmer waters. It was more laterally stratified here, although you do get a sense that this particular station here, which looks like probably DBO 4.3, also had this near freezing water below the sea ice sitting on top of above freezing waters. So there's one interesting kind of anecdotal observation that we had. And then at another station, here we saw this indication with a lot of hits of what looked to be fairly small fish. So maybe one could speculate and say that these could be Arctic cod, and the big fish could be something bigger like a pollock or a Pacific cod. And then there was this what looks to be a fairly non-biological type of signal here in the higher frequencies. And I think that this is the EK80 acoustics showing us the presence of Frazzle Ice. And it even appears that the Frazzle Ice may have incorporated some sediments and settled out on the pycnocline here. You see this band of elevated acoustic returns sitting below the upper layers and the lower layers. That band is right at the pycnocline. And so I think that some of the Frazzle Ice is actually denser than the upper layer, but lighter than the lower layer. And so it's forming this sort of slushy layer right between the two. So again, sort of an unexpected observation, but something that could be pretty interesting to look into in the future. So then we continued our foray south. We actually did the DBO2 line last, but I'll let Jackie talk about it next.
Sure. Just a couple of points I would make, because of Seth's put this in on the Frazzle Ice. I mean, that is a mechanism to bring pollutants or any type of surface sediment contaminants that might be there up into the Frazzle Ice and transported to another area. It's something we've done in other studies for ONR. The other thing I wanted to point out is that DBO3, which is north, he just talked about. One of the nights we were sampling with the Van Veen grab, which is what I use to get animals off the bottom of the ocean, is that we opened up the ice, the moonlight shining down, and then this minke whale decided, well, look at this opening, and it eats fish, you know, so it kind of ties in with some of the acoustic returns that Seth might be saying. Anyway, he did a show and tell, so we all, of course, stopped sampling and just watching this minke whale that shouldn't necessarily be up there in November. And I think that because it's a subarctic species. So, you know, that was some of the interesting findings we found too from the just observations when you're doing your work. DBO2 is this Chukchi Basin. That's down in the middle between St. Lawrence Island and Bering Strait. Like Seth said, we did that actually. We ran over it, went to DBO1 so that to beat that storm that was coming. The important thing is you see on DBO at the top left is the temperature. And you can see that warmer, even on the surface, that's over towards the Alaska coastal water, I believe, over on the right side of the graph. You've got the salinity pretty up and down, though. It's what's interesting on a vertical, not much of a vertical stratification. You can see that in kind of the density, but look at how, anyway, though you can see that the salinity even so was still pretty high at 33 or over at 31. And maybe I have that backward. Is the 0 to 50 close to Alaska? Seth,
no, the little red star over in Russian waters is at kilometers.
Right. Okay, so then I had my description right. You know, so it's just that all of a sudden this is a lot of different type of water masses than normally we see in July. If you look at the lower left, you'll see still a lot of oxygen in the water, and there was some smeared out on the fluorescence on the lower right. So this is an area where gray whales has traditionally been. In the bottom would be these amphipods that gray whales would feed on. We saw gray whales there too in November, and so there was feeding going on. Next. And then this is southwest of St. Lawrence Island. This is where we were lucky to get down to. Again, you see the temperature, and on the left there, you're seeing that pretty warm up and down water. That's the beginning. So that's the water coming up that up on the slope area from the Bering Sea into that region. So we've got like 2 to 3 degree water on that western side of the system. But even though, and then you can see it cooling down towards the as we head a bit north. So on that. The salinity, you can see the high salinity on the left side of the graphic, 33. And then as you get over, and you can see a little bit of stratification there on the salinity. That is why you see some of the patterns in the density. Oxygen, pretty high again up in the surface water. And I would point out the fluorescence, you know, it pretty low overall, but there was a bump up. We had nutrients there, and we did have a bump up in the chlorophyll, and I'll show you some of that.
Oh, sorry.
Next. And I think this goes on to the water column. So when we're there in July, I just point out on the left there, this is integrated through the water column. The highest we found was about 31 milligrams per meter, was a meter squared there. In July, when we're out there on Canadian cruises, it's around 300 to 400. So it's a magnitude less that you're seeing, yet you're still having some chlorophyll in that water column. If you look at the left part, that left graphic in southwest of St. Lawrence Island, that's that little bit of bump up of chlorophyll that you were seeing in the previous ODV graphic on that. In addition to the water column, although it's low, like I said, you have this bump up, you're picking up some of that nutrients and some of that warm water coming up from the deeper Bering, a magnitude less than our July cruises. If you look on the right side, you're looking at the sediment chlorophyll. These are the patterns we get. This provides the chlorophyll. It's settling down pretty fast, gets used by zooplankton, microbials, but there's a lot of organic matter in the summer and it settles down to the underlying sediments. So what you're seeing in the bigger circles is more chlorophyll. It's still half, so it's being used from July to November, but you can see the offshore, the blowout on that lower right there, that's the offshore picking up that anadir water, the little dots near Point Hope there that you're moving in the coastal water system. And then like you can see, you can still see the patterns of that little bump up on the southwest, even in the underlying sediments. So even though it's half of what we're seeing in the summertime, the patterns are still there of where the material settles out to the bottom. And this provides food for organisms that live in the sediments. Next. And then this is the last one. This is a vertical chlorophyll concentration. And basically it's the composite. Like I mentioned, it's an overall low compared to our summer values. But you do see this spatial variance. And so you're going from the left part, so that's 62 degrees north. So now you're looking at latitude and you're going to the right to DBO4 at 72. You can see that some of the water column chlorophyll in that DBO1, this is around the M8 mooring that Phil Stabino has there. And so we have a time series observing going on with that mooring system, and we were able to do the turnaround on that. DBO2 is the Chirikov Basin, DBO3, and you can see where that the chlorophyll, most of it, even though very low values, was sitting down in that bottom region, which is why we have the highest productivity of the shelf system. And then you can see when we moved up to DBO4 in the Chukchi Ecosystem Observatory. So even though low values, you have variance going on from south to north. Okay.
Yeah, and then in addition to the station work, the other primary objective of the cruise was to service some moorings. We wound up recovering 6 moorings and deploying 7. Normally, I try to avoid doing any kind of mooring work at night, but it turns out that in the presence of sea ice, you don't have a whole lot of waves, and that's nice. And we were able to see the mooring floats very, very clearly with spotlight from Sikuliaq. And so in good weather, it's actually seemed to be a great way to do mooring activities. This is a plot at the top showing the time series of the CEO work. The CEO formally started in 2014, and that's all the data from October 2014 to the present. The few years prior to this, data were from a site at an industry mooring that was not too far away from the CEO site. So we're able to extend that time series back a little bit. This was the deployment of the CEO sediment trap, and there's a passive acoustic recorder and a CTD on this mooring. This was going in at the CEO site. This is what it looks like with the ice around it as that sediment trap is being lowered into the water. We got back a sediment trap from the DBO1 region near mooring M8 southwest of St. Lawrence Island with a full set of samples. Catherine was absolutely thrilled to get her 24 bottles back, so that's nice. And we also have a good set of trap material recovered from the Gulf of Alaska over this last year as well, from the GEO, Gulf of Alaska Ecosystem Observatory mooring. So when we finally get back our CEO mooring for last year, hopefully we'll have 3 of them: Gulf of Alaska, Bering Shelf, and Chukchi Shelf, to make a really nice latitudinal comparison of what's being captured by the sediment traps. And that, I guess, brings us close to the end of the cruise. Just a pretty picture here of new sea ice and frozen puddles and frost flowers trying to form, and the low sun on the horizon. And with that, I'd like to thank you all for your attention. Happy to take any questions that you might have for us.
Yeah, thanks, Seth. So I was going to look and see if anybody put anything in the chat, or more than happy to have any questions, just fire away. Look like I can tell you, whoever's gonna say, I can tell you Catherine Lalonde spent the last month here. And so she's been working through a sediment trap, and there's one big difference between the M8, which is in DBO1, and the CEO, is that although she's seeing seasonality in the chlorophyll, there's a lot of sediment, fine sandy type sediments being quite high in those sediment traps at certain time of the year. So there's a lot of resuspension in that area. Okay, Kathy, you're up.
Yeah, well, oddly enough, I want to post a question to Lisa Eisner. Lisa, when you see those graphics of the acoustics and the zooplankton, small fish, big fish, what were you thinking?
Well, acoustics really isn't my expertise, so I mean, it sort of seems like what Seth was showing, but again, I'm not the right person to answer that question.
Okay, sorry, Lisa.
But I can say Lisa has been done, the collections were made for phytoplankton taxonomy and also size fractionation. So she'll be able to have her own presentation about her data.
That should be fun.
Yeah.
And then I do have a question. Do you have anything to describe about the zooplankton?
We did do zooplankton tows. I think we targeted every other station, and we pretty much got them done except along the couple farthest south on the DBO1 line. Because of the wave conditions, we decided to terminate that sampling and not risk the gear. So we missed a couple samples, but we do have some from all four of the DBO lines. What to say about them, I'm not sure I can answer you too well. Jackie, do you have anything to say about?
No, I just vaguely, but I don't know if it's true, if my mind is playing tricks on me, that there were, obviously there were copepods on there, but there were some sites I think had euspauses, but I wouldn't hold that to me. But they'll be processed under Russ Hopcroft's group up at UAF. So we'll have some more information on that.
Right.
Thanks so much, Seth and Jackie. It was a great presentation. It's really exciting to see some data from a season we don't usually get out there and sample. So thank you for sharing that with everyone. Do you know who's doing the analysis of the harmful algal bloom samples, both from the water column and the sediments?
Sure, the water column work that's being done by Don Anderson's group, Evie Vachon there at Woods Hole. So those samples were collected there. And for the sediments, we collected both sediments and also macrofauna. So the sediments also went to Woods Hole, to Don's group, but the macrofauna, the dominant, because the clams and the worms are actually accumulating some of these toxins in them. So we collected those for Kathy Lefebvre and Noah down in Seattle. And so we've sent her those samples, they're processing them, and then Don's group's processing the other part of it. And the eDNA work was done by Matt Galaska's group out of University of Washington. So we'll have some information on that too.
Great, thank you.
I guess a general question back for, Jackie and Seth and any others. I know this cruise happened a little bit opportunistically and late, but you said it was unique to go out into the environment that time. Would that be something that you would want to repeat again?
I sure would.
Yeah, I know, he's a happy boy. Yeah, I would. I think if we could, the ship was great. Could open it up. I think that's the exciting part of it, because you're changing the timing, right? So when the ice is coming back. And I think that, and when it's developing, and I think we're learning a lot by looking at, we need to look at those edges earlier in the spring and later in the fall. So the answer is yes, it just depends on what kind of measurements, doing hydrographic surveys, yes, doing moorings, maybe if it's thin enough ice, yes. And I think Seth was happy with all those, with the development of the sea ice. So I think the answer is yes, I think it would be worthwhile to do. You'd have to decide what type of science could be collected depending on the condition.
Yeah, it's definitely more risky to be doing the mooring recoveries when you've got ice around. We did not recover the NOAA mooring C12
right
in the DBO3 region because it was underneath some fairly heavy ice, and we first would have had to have waited about 8 or 10 hours to get to daylight because the ship did not want to try popping that one in the darkness. And then we likely would have had to have broken the floats and the mooring out of the ice that came up underneath. And so that's an operation that could be, well, 45 minutes if it went just swimmingly well, and it could be 5 hours if it didn't go really well. We just didn't have that kind of time to play with. Similarly, I would have liked to have recovered the CEO moorings. We did not for two reasons. One, they were also under fairly heavy ice. Again, we could have tried to make a hole in the sea ice and gotten it back. But also due to some of the logistics issues associated with spinning up this cruise so late, a couple of our key instruments were not with us. And so last year, thinking that maybe COVID would still have some impacts on our mooring operations for 2021, we programmed all of our mooring instruments that we could to sample for 2 years. And so rather than risking pulling up the mooring and getting it caught under the ice, we think that our fisheries acoustics, our ADCP, our PCO2 sensors were all happily sampling away and will continue to sample away until next year. So rather than disturb those, we just left those in under the assumption that we'll get there in open water in 2022 and pull them back. I think maybe with a little more experience in recovering moorings in the presence of sea ice, we would get a little bit more comfortable with it. But again, I think that we just didn't have the time on a cruise like this to be able to gamble, a half a day to a day's worth of time over one operation if anything went wrong at all.
Yeah, and Seth, there's a question here for Laura Gemery. Is that, are there any, could not be, let's see, other than any standout comparison between Knute's 1960s data? Because he's a physical oceanographer.
I was kind of surprised at, there were some above freezing waters in the Chukchi Sea in that prior cruise as well. So that was, I guess I would have guessed that it would have been all sort of right near the freezing point, but it looked like that's water that had the opportunity to be advected north through Bering Strait. So some things have changed and some things haven't changed. Up in the northeast Chukchi Sea, I think it was more ice covered back then and more and closer to the freezing point in general. But I haven't looked up the ice maps for that year, and I want to do that before being able to really make some good comparisons. But I think it will be worth doing.
Okay, and then there was I think we'll take this as the last question to close up, and that was related to we were able to do the moorings, but I can answer part of this. Anything that was planned for the discovery that couldn't be done, and we left a lot of equipment in Dutch Harbor because we had a vessel. NOAA was going to bring up all the stuff, such as the camera system that was supposed to go into the CEO, and a lot of the NOAA mooring type equipment. One, we couldn't get it up to Nome because the vessel and the weather. You couldn't ship it up to Nome. So there was a fair amount of the mooring operations. The water column work, the hydrography work, the eDNA, the HABs, the benthic work, the zooplankton, that all was able to be continued on with the chlorophyll because we were able to get that equipment and supplies. Some of the supplies had to be shipped again from Seattle because you couldn't get them out. All the chemicals you had to pull in a lot of favors from friends in various parts of Alaska. So it was, it pulled together, but not everything, no. Particularly the mooring activities got reduced.
Also, we didn't get as many stations done as we would have.
Right.
We didn't do anything on the DBO5 region. We didn't even try to get up there, we knew that we were time limited for that.
Right, and the Icy Cape line is another one where actually there's a lot of HAB activity in the past when we've done cruises. So there were those limitations, but what we got up there and the amount of time we had and the lack of sleep, I think we got a heck of a lot done. So I thank all the funding agencies and the people involved. So I think at that point, I think we probably should just, Danielle, I'm going to throw it back to you or Kathy to take on the whatever needs to be done in the rest of the meeting so we can close out by 3.
Yeah, well, thank you all so much. And thanks everyone for joining us so close to the holidays. And before we go, I would like Jackie and Kathy and all of you to join me in thanking Drew for his service to the Marine Ecosystems Collaboration Team. This may be his last webinar in his official capacity with the IARPC Secretariat, and we really want to thank you for all of your work with us. Well, we've really enjoyed working with you, and we'll miss that, and we hope you continue to join our collaboration team meetings as a participant in the future. Thanks so much, Drew. We wish you all the best.
Thank you so much. It's been great working with this team, and I definitely hope to stay active and keep returning to these meetings. And we'll see where the future takes me.
That's great. I do have are we going to do a little announcements through just to update it, because probably all you know, the Ocean Sciences meeting, AG just finished, but the Ocean Sciences went completely virtual. But there are sessions all set up, and we will have Seth and I, Katrin and Lee, have a session on all aspects of the Arctic national and international, and that session is going forward. It's all going to be virtual on that. The other thing is a Synoptic Arctic Survey. NSF has funded, but we're still looking for the ship for that program. It would go from the slope, hopefully in the western side, from the shelf of the Chukchi to the North Pole. We've had other international programs that have already gone out, but there's still some that are going to go out next year too. So I'll give you an update of that when we know more, because there'll be open spaces on the vessel. But I bring it up because we will have Karen Ashton and I have been approved for a town hall during Ocean Sciences meeting. Can post that in the IARP, the Marine Ecosystem Collaborative Team. That will again be all virtual, but we'll provide you the most up-to-date information we have during that town hall. So I guess, does anybody else have any updates they want to say about? Oh, and then we have the North Pacific Research Board's meeting in January, right, Danielle?
Yep, the Alaska Marine Science Symposium. It's not just NPRB, it's a joint effort by quite a few different sponsors. It will be an all virtual meeting again this year, instead of being hosted in person in Anchorage as is typical. And the registration is free, so please look for alaskamaриnescience.org and register to join us for the Alaska Marine Science Symposium the final week of January. And because of the overlap with that symposium, we will not hold our January Marine Ecosystems Collaboration Team meeting. We'll encourage you to join the Alaska Marine Science Symposium instead. Thanks.
The only thing I would update, because I just found out the Arctic Frontiers meeting that's supposed to be in Tromsø, Norway, was just postponed. They're not taking it virtual at all. So that's one often overlaps with Alaska Marine Science Symposium. So it'll be, so wait for a future announcement on that, but it's very much dependent on having in person, and anyway, it's postponed.
Yeah, it looks like there's a lot of things up in the air. But knowing that, I hope that everybody has a good holiday season. We're on the cusp of Christmas and lots of other holidays going on. And Jackie and Seth, really thank you for your time. Really interesting presentation, interesting report from this cruise, and echoing Danielle's sentiments of thank you for Drew for helping us this last couple of years. It's been really a nice engagement, and I hope we get to stay connected.
Great, sounds good. Thank you again for all of that, and thank you everyone for coming today. So unless there's any last announcements or questions, I think I'm going to stop the recording. This will be available on the IARPC website. I think by end of today, actually, we're rushing to get everything done before people leave. But we hope to see you in the new year, and especially at the Alaska Marine Science Symposium.
Happy holidays, everyone.
All right. Happy holidays, everybody.
Happy holidays.
Bye.
Bye.
You.